My name is Ayad. I'm a medical oncologist and hematologist with Cancer Specialists of North Florida. I am very happy and pleased to present to you today Dr. Tony Blau, a dear friend and colleague. Dr. Blau does not need any introduction. He goes by Tony because he's very approachable, very personable, and very much a friend. Tony is founder and CEO of ONCare. It's a platform that I'm sure many of you are very familiar with. He founded ONCare after spending 27 years as professor and physician scientist in hematology and medicine at the University of Washington. And he did lots of research while he was there, and his research studies, they did span a number of topics, including hematopoiesis, gene therapy, stem cell biology, genomics, cancer, you name it. And he's consistently focused on bringing the latest and greatest research advances to patients with incurable disease like myeloma. He also founded the Center for Cancer Innovation while he was at University of Washington. And in that, he brought together a distributed network of investigators to help patients with advanced cancer. He also co-founded the Stem Cell and Regenerative Medicine Institute at the University of Washington. He chaired molecular and cellular hematology study section for the NIH, and he's authored 90-plus scientific publications. Tony continues to infuse an incredible and necessary sense of urgency in ONCare. And in my eyes, he is the ultimate advocate for multiple myeloma research, as he himself was diagnosed with myeloma back in April of 2015. And without further ado, I will give it to Tony, and please take it away. Thank you, everyone.
Thank you, Ayad. Good to see you. Thank you. So in academic medicine, there's a general premise that you should not should avoid hyperbole. But it's my hope that today's talk, in retrospect, will be viewed as historic. And I hope that that I can convince you after my talk that this otherwise hyperbolic characterization is justified. So people that are older like me will remember multiple myeloma as being a really miserable disease. My upfront and close perspective of this came from the experience of my colleague at the University of Washington, Nelson Fausto, an eminent pathologist, author of the main pet textbook in pathology, editor of the major pathology journal, who got myeloma, and it just crushed him with cord compression, essentially collapse of his spinal cord, renal failure, paraplegia, and in an article that his wife published after his death, he suffered more than any other patient that his doctors had ever cared for. Now, I was diagnosed with myeloma just over 9 years ago. I had some hip pain. I thought it was hip pain. I'm not a very good diagnostician. It turned out it would hurt when I press on my pelvis. It didn't actually hurt when I moved my hip. That should have been a clue. But Sibel told me to get an x-ray. For about 3 months, I ignored her, took Advil, got an x-ray, had a big plasmacytoma in my hip, in my pelvis, about an 8-centimeter plasmacytoma. The middle panel shows what it looked like on PET CT. This is a picture taken by Sibel where a set of biopsies were being done on me using a team of doctors and assembled staff that we had assembled for a clinical trial that we had been conducting in patients with metastatic triple-negative breast cancer. And now everyone's attention was turned toward me. Now, with my diagnosis 9 years ago, it was only 8 years after Nelson's diagnosis. But my prospects were very different. And that's because of a plethora of new drugs that have been developed, new and effective drugs that have been developed for multiple myeloma during that intervening period of time. And so whereas the survival of someone that was diagnosed back 20, 30 years ago was incredibly low, and the survival curves are shown to the right of this graph, or to the left of this graph, I guess. It went from less than 12 months to 3 years to now an average survival that probably exceeds 8 years. But with all of those treatment options came a dilemma of trying to figure out which treatment should I choose for me at a given point in my illness. And here, the first decision that I had to make was, should I start with drugs to treat my myeloma, or should I irradiate this big mass in my pelvis first? And as I was trying to figure out my solution to this problem, I decided to use drugs first, and I'm happy to explain why, over beers or something. This is really the first of hundreds of treatment decisions that I've had to make over the last 9 years. And what I most wanted to know is, what did every other myeloma patient choose to do? What were their treatment choices? And what happened to them? What were their outcomes? There are 170,000 of us myeloma patients in the United States. And, fortunately for people like me, it's growing because we live longer, because we have more treatments. But, so what's happening to all of us? If we knew what was happening to all of us, that'd be an amazing data set to have. And it's about 1% to 2% of all cancers. But there was no way to get this information. It wasn't available. And for this reason, I started ONCare. After an initial treatment that included a stem cell transplant, kind of started to put the company together. So ONCare is an online platform for patients, clinicians, and researchers focused on myeloma, where patients register and they sign HIPAA release forms to allow us to access all of their medical records from all of the institutions from which they've received care. And this is by a team that knows a ton about multiple myeloma. We're Myeloma R Us, for the people that might remember Toys R Us. It's all we think about. And then we also have clinicians, both expert clinicians and general oncologists, who increasingly now come to rely on ONCare to provide expert-level care for their patients. Our team includes my co-founder, Ricardo Lopez-Barquilla, who is one of the lead developers of the Microsoft Surface tablet. Mark Walker is an important consultant who's joined us here, the former chief scientific officer at Concert AI. Other oncologists, and Katie O'Brien is our chief of staff, and Jennifer Renz, our project coordinator, and they're shown here. So once we create a dashboard, we are incredibly diligent about the accuracy of the information that we capture, its completeness, its granularity, and its accuracy. It's the kind of data that you would want yourself if you had cancer, if a relative, if your mom had this myeloma, you would want to have data that is supremely carefully put together. And so this shows how it's organized, where you show all of the treatments. Each circle indicates a drug. The colors are iconized. So pretty quickly you get accustomed to the idea that yellow is always bortezomib, red's always dexamethasone, green's always lenalidomide. And then at the bottom you can see the markers, the disease markers that indicate the state of the disease. And so over time, we track the patient's disease over time. Here you can see the disease seems to be under relatively good control. With ongoing maintenance therapy, now it looks like maybe that purple line's starting to rise a little bit. Maybe there's some evidence that the disease recurs. And it's just important probably to explicitly state that despite all of these treatments that work so well, myeloma is still incurable. It's virtually impossible, not impossible, but virtually impossible to completely eliminate the last myeloma cell. And then on top of this, we capture monthly patient-reported outcomes that we layer on top of this. Now, the way ONCare is organized is that the patient can see this, and anyone else who's registered for the platform can see this. We've taken away the patient's name, we give them a unique identifying number, and most importantly, the patient's doctor can see this. And this saves the doctor time. So as we embed this platform in a doctor's practice, this whole chronicle of a patient's disease from diagnosis to present gives a doctor a view of what's happened that is accurate. It doesn't have gaps. I mean, sitting with doctors sometimes who are trying to figure out, and having been in this position myself many years ago, trying to figure out what's happened to a patient, you're trying to figure out, okay, have they received this drug? How much did they get? This was 3 or 4 years ago. I can't even remember this for myself. And you have just a few short minutes to make this decision for your patient. So here, you can come, look at this, get this comprehensive view that is just not available anywhere else, and quickly and accurately understand what's going on in your patient. So after we embed a physician's practice to use this platform, this will save you time in taking care of this patient. And if it doesn't, let me know and I'll buy you a cup of coffee. And give us a little bit of time to get it working well, but this will really help in caring for patients. And we capture the records from everywhere where the patient's been seen, every institution. We pull it together so you don't have these gaps of what happened when they got a stem cell transplant and things like that. We capture everything. Again, just like you would want for yourself or your mother. We've done this now almost 1,000 times, 930 sometimes to be exact. So each patient super carefully curated. And what that allows you to do is to compile this information so that now you can see across 930 patients which line of therapy did they receive, what was the content of that treatment, how long did they receive that treatment for, what was the response, and what order was that treatment given relative to all other treatments that the patient received. And based on that visualization of those 900 plus patient histories, we can begin to generate reports that we call myeloma map reports that describe the treatment choices of other patients like your patients. What were the treatments that they chose? And what were the outcomes? Another thing that's a key part of the way we work is that every new patient dashboard is reviewed in a virtual rounds that's similar to the type of rounds that I made at Fred Hutchinson and the University of Washington, except that rather than walking down a hall with a bunch of people in white coats following you, going from one patient's room to the next, we're gathered around a patient's dashboard virtually. Patients presented by one of our medical data specialists. We have input by senior oncologists. And we're looking to vet each dashboard to make sure that our understanding of what has happened to the patient is accurate. Is there anything here that doesn't make sense? Are there any outstanding questions that we have about this patient? Again, if you have cancer, this is what you want. You don't want these questions about what happened at a particular time. You want to really dive deeply into understanding what's happened to the patient. And then we provide, and then we can provide input that can be posted on the patient's dashboard. And ultimately, from this process, it allows for patients to have access to care, to treatment ideas that are not available otherwise. And this leads to another key point that we found. Probably the most important thing that we found is described in a publication in an abstract from 3 years ago at the hematology meeting, the major hematology meeting at ASH, where we found a tremendous amount of variation in the way that patients with myeloma are treated in real-world settings. So it's kind of mind-boggling. And the implication of this is that if you have cancer and myeloma as an example, your amount of remaining life is very dependent on the approach that's been taken to treatment. And so if you've been treated with one approach A here, you might have lived for a particular period of time before you died. Treated with approach B, you might be alive today. And treated with approach C might have been a really bad choice, all for the same patient. And so this leads to lost time for the patient with family and loved ones, needless suffering, of course, and wasted resources. Another way of depicting this variation can be seen in this type of plot called a Sankey plot. And Mark Walker, who's here in the audience, built this for us based on the results that we've had from our patients. And on the left, you can see LOT stands up for line of therapy. So the width of each colored bar reflects the number of patients who received that specific line of therapy. So for first-line therapy, you can see the top bars where you have the most patients, and then it becomes fewer and fewer until the bottom is an other category where those patients have received a frontline therapy that almost no other patient, perhaps one other patient or many times no other patient has received. And then when you move to the second line of therapy, now you can see terrific amount of scatter in the second line therapies that have been chosen for these patients. And so it's an incredible level of heterogeneity, so that by the second line and certainly by the third line of therapy, every patient is on a unique path that no one else is on. They're getting something different than anyone else has received. But why is this? Why would this be? And it's kind of obvious why this would be. You look at the guidelines that are available to oncologists to deliver care to patients with myeloma. The NCCN guidelines are key guidelines. And you look at the choices that you have for first-line therapy. If you're a transplant candidate or a non-transplant candidate, the choices are incredible. And what's an oncologist supposed to do where you might only see 6 patients a year? It's not like lung cancer. It's not like breast cancer. These are patients that are coming with a relatively rare disease. They're coming to you for treatment, and you have so many choices and very little guidance about what to do. So the tools suck to deliver expert-level care to patients. And if you think about patients out on their own with their oncologist crossing their fingers, hoping for the best, it's like everyone for themselves. Best of luck to you, but you're just trying to navigate your way through this terrible disease with the best efforts of you and your oncologist. So to address this issue, we developed with great input from IED and Tara and others in the heme committee at QCCA and now ONCare, have developed a clinical pathway for patients with multiple myeloma that's being released today. And it has a number of features that are described here. We have 100 copies of a document that contains this pathway that'll be available for people here to take home. It's a 27-page document. But it has a number of features associated with it that are described here. There are 2 ways that the pathway can be accessed. One is for the patient to register for All4Cure. And there, after the patient's registered, there's a link to the 27-page PDF. But one of the benefits of being in All4Cure is that after the patient's registered, now we can track the patient with you over time and surface the relevant bits of the pathway that the patient and the doctor would want to consider at that particular time point of the patient's treatment. So we're chunking it out so that you can see, disease markers went down. We're interested in trying to achieve MRD negativity. Now might be a time to consider a bone marrow with clonoSEQ testing, for example. So this just-in-time guidance is really important. I should emphasize This pathway is very heavily focused on newly diagnosed patients with multiple myeloma. And so what I would like to instill in everyone's head here is, if you have we're interested in all patients with multiple myeloma, but if you have a newly diagnosed patient with multiple myeloma, please think ONCare. Newly diagnosed myeloma, ONCare. Get the patient on the pathway. The other way to get the pathway is to download it from our website, or contact Katie at allforcure.com, and we'll send it to you, the PDF. And we feel like this is a great pathway, can help a lot of people, and we want to extend that and distribute that as widely as we can. One key aspect of the pathway is that it's forward-looking, in that we have embraced the concept that it is desirable to achieve a state of MRD negativity, measurable residual disease negativity, in patients with myeloma. Now, there's not a uniform consensus by any means among the myeloma expert community that that's the way patients with myeloma should be treated, but this pathway is focused on achieving MRD negativity for patients that are on the pathway. And that's because here it's super clear that patients who achieve MRD negativity live longer. Now, what's still debated is sometimes the extra treatment that's required to achieve MRD negativity, could the toxicity of that extra treatment counterbalance the benefits of achieving MRD negativity? And that's not yet completely resolved. But within the pathway, we provide a whole set of guidance about antibiotic use and other measures that you take to minimize the risk of complications. So I think we can, by following the pathway, we can mitigate concerns about infections and other complications that could happen as a consequence of using the drugs in this pathway. I've been incredibly focused in my own treatment on trying to achieve MRD negativity. And, there's the data that shows that you live longer if you're MRD negative, but let me tell you for a moment why I embrace this philosophy so strongly. And that's because every cell in a cancer and in a myeloma cell is different from every other cell. Every cell is unique. And we don't really think about that much. But every time a cell divides, there's an error rate in DNA replication of about one base pair per billion bases. And so with each cell division, that amounts to about six changes out of 6 billion. Wouldn't really think that matters that much. And they tend to be random across the genome. But what that does is it makes every cell slightly different from the next. And under normal circumstances, that doesn't matter. But under selective pressure, if you give a treatment now, occasionally, some of those changes might confer upon a cell the ability to resist treatment that every other cell is susceptible to. And so that combination of variation and selection allows a small population of cells to survive. And of course, errors in DNA replication can go way up in cancers. So this 6 base pairs for every cell division can increase massively in the context of cancer. But this concept is not widely appreciated, that you're not just combating myeloma, you're combating millions of individual, unique myeloma cells with every one of your treatments. And the other thing I think about as a patient is, I'd love to have a little finger prick, or better yet, a little thing like a pulse oximeter that just tells me it's a myeloma cellometer, where it tells me how many myeloma cells I have in my body. That's what I'd love to. I'd love to track that day in and day out. I'd measure it 10 times a day and just track that over time. Of course, that's not possible. But you can think about, in general principles, roughly how many myeloma cells might there be in my body? And to think about that, there are about 30, there's estimates of how many total cells are there in our bodies, in the human body. And it's been estimated that maybe they're on the order of 30 trillion cells in the human body. And then if we think, okay, at the time of diagnosis, when I had this big plasmacytoma and myeloma in my bone marrow and stuff, I probably had hundreds of billions of myeloma cells in my body. And now let's say you get treatment and you knock that down 1,000-fold. That's a great response, right? 99.9% of the cells are dead. But you still have hundreds of millions of myeloma cells at that stage. And even when you're MRD negative, and MRD negative meaning there's this whole thing about it's 10 to the minus fifth or 10 to the minus sixth. I think that's a boring debate. If you look at one myeloma cell in 1 million bone marrow cells, and you can say there's no myeloma cell per 1 million bone marrow cells. Gosh, that's great. 1 million cells, no myeloma. The problem is, if there are 30 trillion cells in our bodies, I guess maybe there are 1 trillion in the bone marrow. And so if you tell me that I have less than 1 in 1 million, I could still have almost 1 million myeloma cells left that remain undetected. And I think it's probably a lot closer to that than to zero. Sibel would like to think it's zero sometimes, but I think it's closer to 1 million than zero. And so the overall idea is I want to minimize the population of cells so that I can reduce the number of cells that might acquire the mutational content that allows it to grow back and cause problems for me someday. And if I can do that with millions, if I can get it down to that million, that's 10 times better than 10 million, where the likelihood of one of these cells creating a problem goes up. So this is, it's not a randomized controlled trial or whatever, but this is the reason that I've decided to really try to focus on achieving and maintaining MRD negativity. And we have some simple videos about MRD, clonoSEQ testing in videos on the All4Cure website. So this is where the historic part comes in, I think, is that we're implementing this clinical pathway across the ONCare Alliance. You guys are the largest network of community oncology practices in the nation. And just a little bit of an editorial comment about that is I think you've come here by choice. It's no one that's forced you to join this organization. It's a coalition of people that are in control of their own fates, want to do new things. I think that's a very important feature. In total, there are over 800 providers across this network. It turns out that my estimate of the total number of myeloma patients was a little high. Aaron Peavy House tells me that there are about 6,655 patients with myeloma who are probably alive today across the network. And that's knowable because there's a common normalized electronic data warehouse that allows that question to be addressed. Okay, so think about this for a moment. Think about a network of doctors who treat myeloma in broadly similar ways. And here in this trial, we're going to test whether implementation of this pathway leads to better outcomes. So we're comparing patients that are treated on pathway versus off pathway. This will be a retrospective analysis conducted every 6 months with the goal of determining whether pathway implementation is associated with better outcomes. So imagine a world in which 800 oncologists decide to treat myeloma patients in broadly similar ways, in optimal ways. And in the pathway, we have these optimal initial choices, but relatively few optimal initial choices. Dara-RVD, plus or minus stem cell transplant, Dara-RD, and isatuximab regimens as well. So let's just imagine everyone agreed to treat their newly diagnosed patients with myeloma in this way. And then let's imagine that the first-line therapy doesn't work anymore, and you move to a second-line therapy. And let's imagine that we choose what we think are the optimal second-line therapies. And we just agree collectively we're going to treat patients in this way. The power that this network of oncologists can bring to understanding what works and what doesn't in patients with myeloma is unprecedented. There's nothing in medicine that has ever happened that would provide the kind of information that you guys can provide by working together, working collectively to figure out what works and what doesn't, delivering optimal care to your patient, while at the same time contributing to a learning network that gets better over time. You will learn together what works best. Together, you will figure out what the best treatment for multiple myeloma is. And the whole idea is that cancers can't learn from each other. My myeloma can't learn from any other patient's myeloma. But cancer patients and their doctors can learn from each other. This is a hidden superpower that we have not begun to exploit in treating patients with cancer. And together, the ONCare Alliance can do this and make progress in cancer care that's unlike anything that's been accomplished today. And so, whereas the other picture showed people scattering in a riot, here, by working collectively, we can change the world. And so this is just, and then this is just saying by through this constant learning, seeing what works and seeing what doesn't, we'll figure out which second-line therapy is best after a given first-line therapy for a given population of patients. So this pathway is a living pathway. It will get better. Right now, patients' experiences, all of that treatment, all of that expense, all of that suffering is lost. It goes out into the ether. We get nothing from that. By working collectively, we can together figure out how we take steps toward curing myeloma. As part of this, we can create a new framework for clinical trials. So this extends to existing therapies, approved therapies, but also clinical trials. Imagine together how quickly we could enroll patients on promising clinical trials and figure out what works and what doesn't in a much faster, much more democratized way than is possible today. We've taken a baby step in this at ONCare by working with four ONCare practices to enroll patients with multiple myeloma. 200 patients are a target. We've enrolled 120 patients right now. Key aspect of this is that ONCare is a single clinical trial site. And this merits its whole other discussion, but the idea is to create a framework that would allow a patient to participate in many different types of clinical trials just by going to see their doctor. Getting treatment, we collect all of the information with incredibly diligent care and allow clinical trials to be executed in a much more rapid, much more efficient, less costly, and more democratized way than is possible today. And then this is just a little advertisement from an abstract, a poster that we had at ASH last December. So please consider registering yourself for ONCare. We have people on standby that can help you do this now. at your beck and call. Please consider asking your patients with myeloma to register for ONCare. We welcome all myeloma patients, but especially newly diagnosed patients. And here's a really important point. Getting this implemented at the point of care with an oncology practice has all sorts of considerations where we really need your help and your partnership about how to do this correctly. I can tell you, we have figured out ways to do it incorrectly. We've done things that we've messed up in trying to navigate the communication between the patient and the doctor, and doing that in a way that is helpful rather than just being a pain in the ass. We have made mistakes, and we're learning from them, and we're getting better, and we're committed to making this work really, really well so that we are empowering an oncologist to deliver expert-level care. But we need your help to figure out how to really make this work well. And those are my comments. Happy to answer questions.
Tony, first of all, thank you for that very passionate presentation. I think your passion does get across to all of us. Also, thank you for sharing your own journey. I think this is extremely powerful, and I think it would touch many people here. I had two questions, and these are more, really operational, if you will. So the first one pertains to the sort of the pathway concordance trial that you mentioned, and if I heard you say it's retrospective, it is retrospective in nature, right? So for all of the ONCare practices, do we have to do something to participate, or is it automatic, or when does this start?
Yeah, thanks for that question. So if it's okay, I'll address that first. You can ask the second, because I'll forget otherwise. Of course. So we're working very aggressively to get the pathway implemented as widely as possible and as effectively as possible across all ONCare practices now. And then the trial that we'll do is a retrospective trial where we will have access to the ONCare database, and we have, with Mark Walker, designed a way of identifying patients who have been treated on pathway versus off pathway over the preceding 6 months, and then we'll fill out case report forms for all of those patients. It's a total of 450 patients, and then every 6 months we'll update that so that we're trying to take advantage of a natural experiment, if you will, for looking at patients that have been treated on pathway within the ONCare Alliance versus patients that have been treated off pathway, and then looking to demonstrate improved outcomes from patients. So there's nothing to do now except get on AllForCure. Get your patients on AllForCure. If you have a newly diagnosed patient, think AllForCure.
And the second question was actually in that regard. Now that you have over 1,000 patients in AllForCare already, so for those of us that might just be getting introduced to this and trying to offer participation to our especially newly diagnosed or currently being treated patients, any words of wisdom, any lessons that you learned, in terms of how we should present it to them? It's like, what's in it for me? Or from a patient's perspective, and privacy concerns and this and that. So any nuggets that you might have for those of us that are new to this?
Right. Those are great questions. So I think there are two categories of patients. There are maybe 20% of patients that are in the nerd kind of category that really love these graphs. But if I ask my, brother who's a Methodist minister to look at my dashboard, it's like he says it looks like gobbledygook. So the dashboard's really tailored for the oncologist. And the patient should register because it helps the oncologist see everything that's happened, learn from that experience, and deliver better care. Every patient for the dashboard, every patient's identifying information is removed, and we keep the patient's identifying information separated from everything that's in the dashboard in our databases. So we've taken great care to preserve the confidentiality of patients. When I, some patient that matters to some patients, for sure, to make sure that we're diligent about that, and we're very diligent about that.
So again, thank you for a very passionate presentation. And I have two questions. Does this all for cure only for the myeloma, or it also has a smoldering myeloma?
We also
who are not on the treatment.
Yes, we accept patients with smoldering myeloma. Thanks, Prabita. We accept patients with smoldering myeloma. Even when patients with MGUS want to come on, we track them as well. So yeah, we also take patients with those smoldering myeloma. So far, we don't have a lot of interest from MGUS patients, but yeah, we take smoldering myeloma as well.
And is there any collection of the myeloma cells for these patients?
So there's no standard collection of myeloma cells from patients. We do have a clinical trial that can be that patients can agree to through a consent that's done through ONCare that allows biospecimens to be collected. But that's that's a part of a separate trial that patients can participate in, and we employ that on an as-needed basis.
Dr. Grass? Great job, Tony. So I was going to say, Ajit, to what you were saying, patients, we all have patients that the first thing they ask us at their visits are, what was my M protein? Or my M spike? What were my light chains? So having this tool allows them to see just that. And so what I was going to say is, you have those patients, right? We all do. And so I think that, the way you explain this is, hey, how would you like to, be able to actually see all of your information, all your data at a bird's eye view and be able to really follow everything. And I think they love it, right? Just the same way they love seeing their MRD. They understand that more than they understand a scan. So I think what you've provided here is excellent. I mean, we're not all experts. I see non-Hodgkin's lymphoma. I mean, yes, I understand myeloma, but to be able to have something like this is just, it's amazing for what you've done for our patients. So thank you for that.
Thanks, Jer. I think, as I tried to emphasize during my comments, we're really focused on saving the doctor's time as well. So I think the patients, but we're committed to making it very clear what's happened to the patient from diagnosis to present. You see trends that you're not going to get from just looking at the last few lab values. And and imagine you have a new patient coming to you. You're going through how many pages of medical records to figure out what's going on. We show you everything in a really very, very quickly digestible way.